All right, good morning. My name is Charles Large. I'm the CEO for Autifony Therapeutics, which is a private biotech based in just north of London. We were founded about 15 years ago as a spin-out from GlaxoSmithKline, where some of the technology I'm going to talk about was originated. We are a company that's focused on the development of small molecules for the treatment of CNS disorders, focused on ion channels, which is really sort of shown in this diagram here. Today I'm going to talk about one particular clinical stage program, which is our KV3 targeting small molecule program. Now, the mental health crisis is something that I think we're all very familiar with. As by way of introduction, mental ill health now represents the single largest driver of disability in the UK, and I'm sure similar statistics can be counted for other territories, including, of course, the US. And, of course, although some of these disorders are often not fatal, they rob patients of their lives. Incredible unmet need in terms of treating these disorders. Current treatments are available, but of course, as we well know, they tend to be symptomatic. They tend to be producing side effects which simply add to the burden of disease. Now, one of the big obstacles over the last decades to the discovery of novel targeted therapies for neuropsychiatric and neurological disorders has actually been having a system of diagnosis, a system of classification that tells you something about the underlying patient's pathological needs. That's not the case with current existing diagnostic approaches such as DSM-5. And to illustrate this point, these are some data from the BSNP consortium. There's an American academic consortium that set out to characterize patients who had a psychotic episode using a range of neurophysiological, neuroimaging, and cognitive biomarkers. And this graph just shows one of those particular biomarkers, which is a neuro-EEG biomarker. Now, what they did was recruit many thousands of patients. This shows a sample of those, and deep phenotype them using these biomarkers. And then ask the question, do these biomarkers differ in patients with schizophrenia, patients with schizoaffective disorder, or patients with bipolar disorder, as shown here? Now, what they saw was actually no. If you group the patients on the basis of these classical diagnoses, you see nothing compared to healthy controls. So what they did was throw away the DSM, take all of these patients and get a computer algorithm to sort through and understand where are the opportunities, where are the differences across these different patients. And what they saw was the computer managed to pull out three very consistent, what they've now called biotypes. So these are clusters where you see biomarkers essentially defining the pathology that these patients have. So biotype 1, for example, shows this low EEG profile, so hypoactivity within the brain. Biotype 2, the opposite, hyperactivity, and then biotype 3 actually somewhere in the middle. Now, what's critical about this finding, which has been replicated now with further repeat studies, is it allows some actionable approaches to targeted drug therapy. And this is where Autifony has really been focusing its attention over the last 15 years. Can we identify these biotypes? Can we identify the pathology underlying it that allows you to say, okay, I have a drug that treats hyperexcitability, in this case, and therefore is going to treat patients, not just patients with a formal diagnosis of schizophrenia, but across these diagnostic criteria. And that's essentially what we've set out to do. But the other element you need for this to be able to do that properly is understanding what's behind that hyperexcitability. So what are those biomarkers telling you about the circuitry in the brain, about what's going wrong, that gives you those actionable insights to treatment? Now, what we do know, if you look at these canonical structures of human cortex, you have excitatory neurons, you have inhibitory neurons. Inhibitory neurons are relatively few in number, but incredibly important. They play a critical role in synchronizing the activity of brain circuits, of maintaining inhibition, maintaining the balance between excitation and inhibition. What's also been shown through the literature over many, many years is that these neurons are incredibly vulnerable to different challenges. So adverse challenges such as genetic causes in the case of neurodevelopmental disorders like Dravet syndrome and epilepsy syndrome, but also disorders like schizophrenia, which come from probably a mixture of genetic and environmental factors. Loss of function of these neurons leads to an hyperexcitable circuit over here. When this happens and the degree to which this happens will determine the severity of the symptoms that actually present in the clinic. So in the case of Dravet syndrome, which is a genetic disorder where these interneurons are impaired by a fault in the sodium channel, you get a very catastrophic epilepsy phenotype. In more milder situations, such as perhaps schizophrenia or neurodevelopmental disorders like Fragile X syndrome, a loss of function, an impairment of function of these gives rise to cognitive and sensory dysfunction. Now, what can we do about all of this? Well, within Autifony, what we've been doing is focusing on the Kv3 ion channel, which is specifically expressed on these interneurons. And what we've shown over the last 15 years is that by enhancing the activity of these Kv3 channels, we can restore the function of these interneurons in a number of different challenge situations, whether it's an epilepsy situation or indeed in situations related to schizophrenia. And a range of these disorders really is shown on the right here, where we've evidenced both preclinical and now clinical, demonstrating that modifying the activity of Kv3 channels with our positive modulators can produce benefits in these disease areas. So a fantastic opportunity both in the rare orphan space, neurodevelopmental diseases, but also in broader diseases like schizophrenia. So where are we with our progress in this program? So we have a number of compounds, some of which we brought into the clinic. 206 is our optimized clinical lead Kv3 positive modulator. It has FDA and EMA orphan drug designation for Fragile X syndrome, which we think is an index opportunity to demonstrate the efficacy of this mechanism in this sort of patient population. And that compound is now phase 2 ready and ready to go into a clinical trial in that patient population. We also have a number of compounds at the candidate stage, which have long IP. They have slightly lowered predicted human dose, and therefore we think are more optimal perhaps for these broader indications such as schizophrenia and perhaps the early phases of Alzheimer's disease, where again we're looking to restore function of these interneurons. So we are currently in partnering discussions for this platform. So Autifony has actually grown quite considerably over the last 5 or 10 years, and we're now putting a lot of our efforts into some of our earlier stage preclinical programs. That's what we're good at. That's what we set out to do when we set up the company. We've established collaborations with Jazz Pharmaceuticals, collaborations with Boehringer Ingelheim, and this is really where we're looking to increase and expand our activities actually. We're not so much a clinical development company. So what we are looking to do at the moment is now partner our Kv3 program with a company that has good clinical experience, good clinical development experience to bring forward and really find the true optimal exploitation of this novel mechanism using biomarker-led studies. So to finish up, Autifony's Kv3 assets have been developed to take this challenge of providing targeted, rational treatments for CNS disorders, making use of biomarker-led clinical trials to identify and stratify patients to get conclusive evidence in those trials. And please, for further information about this or for interest in talking to us about partnering, please feel free to contact me. Thanks for your attention.